Alright, so this is part one of our EOC reviews. We're going to start on the very first page of your packet. You should have received a packet already in class, and if for some reason you weren't here and didn't receive a packet, then you should have, I'll also be emailing this to you as well, so you can open it in an attachment and fill it out at home, print it out and fill it out at home. But for our review, what I'm going to do is provide you with the first couple letters of the word. For some of the sections, I'm going to go ahead and define it for you or give you the information. But for other parts, you are going to get a little hint, and then you'll be expected to go back into the unit, back into the notes, and fill it out on your own. What we're first going to start with, we're actually going to start with Unit 1. And in Unit 1 of our notes, we looked at organic molecules, and we also called these macromolecules, large molecules. Let's really quickly define the word organic. Organic molecules are going to contain carbon, hydrogen, and oxygen. And they do have to have all three of those elements. We had four main macromolecules. The first was carbohydrates. And carbohydrates are mainly going to be used for quick energy. And also sometimes structure, like in plants. The subunit, another word for subunit is also monomer. And again, you do want to know all these vocab words. Even if two words mean the same thing, you should be able to associate them. So subunit and monomer means the same thing. So therefore, macromolecule, or it starts with a P, polymer means the same thing. For the monomer of a carbohydrate, it actually also starts with the word mono, and it really means one sugar, because a lot of times carbohydrates act as sugars in our body, and that's how they provide that quick energy. So the word for that is monosaccharide. Then our second macromolecule is a protein. And a protein is going to have several different jobs. It's actually going to have 6 jobs. And that could be acting as a pigment to absorb light. Pigment, I'll write for light. Acting as an antibody. Or allowing us to transport different things within the body. So pigment for light, if you think about the chloroplast, there's an enzyme called, there's an enzyme called chlorophyll that absorbs light. That's how the plant does photosynthesis. Antibodies are going to help you prevent infections in your body, prevent illnesses. And then in transport, well, in your body, your red blood cells, proteins in your red blood cells will help you transport oxygen. There are 3 more jobs, and you do want to go back and make sure you fill those in. For the subunit starts with an A. Do you remember what it is? 2 words, both starting with an A. That's actually going to be your amino acid. Oops, I did a very good job there. Amino acid. Let's stop for 1 minute, because we did say that organic molecules have carbon, hydrogen, and oxygen. And we just want to remind ourselves of what elements are in these macromolecules. For carbohydrates, there's only carbon, hydrogen, and oxygen, a simple organic molecule. For proteins, it's a little more complex. We'll actually have nuts. Do you guys remember that? The 6 essential elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Then we have our lipids. Lipids also are known as fats. Another word for them. And that also contains just carbon, hydrogen, and oxygen. What are fats used for? Well, they are a source of energy, but they are going to be long-term energy. And they're also going to be there for insulation, such as the fat in a bear, keeping it warm. And also for cell structure. What part of the cell is made of lipids? What really important part? The main kind of container of the cell. Well, the cell membrane is made of phospholipids, and that's really important to remember. The subunit for a lipid is actually called a triglyceride. It's made of two parts, two main things, a glycerol and 3 fatty acids. So you can see triglyceride. You can also see fatty acids. And that would be the monomers of your lipid. Then you have your nucleic acids. Nucleic acids contain information in the form of the monomer nucleotides. And the main function, again, is information. Your genetics. Here you'll have the elements ch, na. Okay? You won't have the sulfur there. Then here what you'll have is your specific molecules, examples of those 4 macromolecules. I'm going to help you first by talking about their function, and then I do want you to go back and think about what subunits are going to compose or make up those molecules. So you have starch. Starch is actually going to be a way that plants store extra parts. You'll have cellulose, and this actually is a structure or structural molecule that's going to help make the plant's cell wall. You'll have insulin, and insulin is going to help regulate blood sugar. You'll have glycogen, and glycogen is actually a way that animals store extra carbs. Glucose, remember, is a molecule, an energy molecule, or can be used for energy. In respiration, you'll see that molecule. Enzymes are going to speed up chemical reactions. Hemoglobin is going to carry oxygen, and that's inside of the red blood cells. Then you'll have fats. Important fat is actually going to be a phospholipid, which is going to make up the cell membrane. You'll have DNA, which has all of your genetic information. It's going to be stored in the nucleus and tell your cell what proteins to make. So info to make proteins as well. Then you have your RNA, which are molecules that will actually help build and code for proteins as well. So what you should do is you should go back and fill in your subunits or your monomers for all of these different molecules. And you will have all these in your notes. The last thing I wanted us to look at today is going to be your tests for these 4 molecules. And so here you have starch, lipids, monosaccharides, and proteins. These are all things that we can find in those 4 macromolecules above. Here we're talking about types of tests. They're also known as indicator tests. And remember that our indicator tests changed color? In presence of that molecule. So starting off with starch, which macromolecule was starch? It's actually a type of carb. And there's actually two carbohydrates in this list, our starch and our monosaccharides. Starch is a big carbohydrate, or also known as a polysaccharide. And then you have your smaller carbohydrate, your monosaccharide. For starch, our test actually started off as a black, it actually starts off as a red-orange. And if there's no starch in your sample, then it's actually going to stay red-orange, be a negative test. But if it's positive, it'll change to a blue-black. So it changes to blue-black if starch is present. For our lipids, we're going to use a little piece of paper, and we're going to test it. If it's negative, the bag stays brown. If it's positive, the bag actually becomes a little clear, a little transparent if you hold it up against the light. For our monosaccharides, we're actually going to start off as a blue color. There's no simple sugars there, it'll stay blue. This is the one where we'll have to use heat. And after we use heat, if the monosaccharide is present, we'll turn to an orange-yellow color. And then the last test is for proteins. And in proteins, we do also start off as a blue color. And to help me remember what color it turns to, protein starts with a P and actually turns a pink or purple color. No heat required here. I want you to go back and figure out what were the names of the tests and fill those in. Okay, that's all.